I wanted to give you an extra slide about this whole binary, decimal, hexadecimal thing because I feel like I made such a mess of it yesterday. So I'm going to walk you through this. This does not appear in your PDFs, so I've added it into the first set of slides so that next year when I reuse this material and reorganize it, I'll have the slide in place. Base what, I've called this. One of the common ways that we talk about binary and hexadecimal and decimal number systems is as base 2, base 10, and base 16. So when you see them in math class, that's the language that we use to describe these. So this base terminology has two meanings in a way. The base of numbers defines how many symbols are usable in each place. So in decimal, we have 10 different numerals that we can use in any one spot within a number, 0 through 9. Okay? In binary, we're working in base 2, and there are only two numerals rather, that we can use in any position within a number, 0 and 1. In hexadecimal, we have 16 different numerals that can appear in any one position. Now, because our normal number system just uses 0 through 9, we have to have some more symbols to build us up to 16 values in hexadecimal. And people have decided that the simplest way to accommodate that is to simply use letters, A through F. So A through F in hexadecimal come after 9. So 7, 8, 9, A, B, C, D, E, F, 10. That's the weird part. So that brings us to the second part of this. The base also defines the exponential increase in weight from right to left. That sounds complex, but you deal with this every day. If I give you the number 384, which numeral of 3, 8, and 4 has the most weight in determining the value of that number? The 3 does. And the 3 gets the most weight because it's the leftmost number. It's 2 positions to the left. You've got the way that we used to teach this is to say we've got the 1's position. That's in 384. That's 4 units. In the 10's position we have an 8, and so that 8 has 10 times as much weight as the 4 does. So it's worth 80 instead of 8 by being in the second position. And then the 3 is sitting in the hundreds position. So it has 10 times as much weight as the 8, which had 10 times as much weight as the 4. So that's base 10. But when we're working in binary, to be in the second position over is not worth 10 times as much as in the units position. It's worth 2 times as much because it's base 2. There are just 2 symbols. So I've got the numbers here from 0 to 16 shown in decimal at the left. And you can see that after we use the symbol 9 to represent 9 units, we go to 10, which means we've run out of this dictionary of possible numerals. We've hit 9. And now we're going to roll it over to 0 and add a 1 in front of it to indicate that we've rolled once through this entire set of numerals. So you don't think about that. You just say, well, of course 10 follows 9. But 10 only follows 9 in a decimal base 10 number system. 10 follows 0, 1 in binary because there are no digits, there are no numerals after 1 in the binary system. You have to go back to 0, which means you have to push a 1 over into the next position over. So that next to last bit in binary is worth twice as much as the last position. And the bit 2 over has twice as much value as this one, which had twice as much value as the one to its right. So hopefully that helps to explain a little bit better how we make this adaptation among binary and decimal and hexadecimal. It's not uber critical to your understanding of the rest of bioinformatics. This is just a computer science concept and a math concept that shows up all over the place. So I hope that it's useful to you to have taken a quick moment to look back on it. In retrospect, including it, expecting that to fit alongside ASCII as I was trying to explain character to number mappings was probably a mistake. But this is still a very fundamental computer concept that I would like everybody to understand. So with that said, I'm going to bop back over to the slides that you should have. I think I've made just one correction, like to add a title to a slide. But otherwise, these slides are just as they appear in your PDFs. So the discussion today is about DNA sequencing, mapping, and variant detection. Along the way, stuff like GWAS, Genome-Wide Association Studies. A few asides about green tea. All right. So our overview is going to start with a little discussion of FRED, which is a way that we represent very low probabilities with negative log scores. This is an approach to evaluate the quality of individual letters in a read, individual base calls. We talked about that this morning. We're going to discuss how it works here. We're going to talk about bowtie, which is an approach that we use that makes use of the Burrows-Wheeler algorithm for aligning reads from a sequencer to a reference genome sequence that we have. And then we're going to talk a lot about genome-wide association studies and admixture structure, stuff like that. Should be a lot of fun. So I made 3 different diagrams that try to show kind of the active areas of bioinformatics research that allow us to slot the discussion that we're going to have over these 8 days into these 3 diagrams. So this is the first of those 3. I was trying to ask, how do the, are we a little out of focus? Still a little bit, but that's all right, I'm going to deal with it. This is a wildly complex chart, and if you think about how many papers underlie each of these developments in bioinformatics, you'd really get lost trying to assemble a bibliography that covers this complex a set of things. My goal here is to give you a roadmap of all of bioinformatics. So treat this as roadmap 1, and it's going to dictate what we discuss for the remainder of this week, days 2, 3, and 4. Today is day 2. We're also going to have a roadmap built around structural biology, proteomics, and metabolomics. That's one that will show up next week. And we'll have one other roadmap about pathway and network analysis, biological pathways, biological networks. That will show up really only on the last day. And we have one day that doesn't really fit onto any of these diagrams. That's a day built around image analysis and flow cytometry. That is Wednesday of next week, if I recall correctly. So we're going to try to orient ourselves according to these maps. As I said, the rest of this week is shown in this one diagram. It's principally a discussion of genomics, and it's principally a discussion of high-throughput sequencing. But, in fact, microarrays can be used for a lot of these purposes if you're not willing to go the more expensive route of sequencing. So today, we're going to talk about the problems of starting with sequencing data or microarray data, base calling against sequencing data, read mapping, figuring out where those reads go in a known annotation, the detection of sequence variants, admixture analysis, which is quite necessary when you have a stratified set of people taking part in a genome-wide association study, and GWAS itself. So all of that's going to fit into one day. These are each topics that could be expanded into a whole class in their own right. Getting only 8 days to talk about bioinformatics is what's available to us, but I used to teach this material across an entire semester, and that was very superficial. So keep in mind that most of what we're going to try to accomplish through these lectures is give you bookmarks to say, I remember we talked about this in class. There were citations in those slides. I can look at that to learn more about what's going on here. So bear with me, even though we know that this is relatively superficial. I believe I have a quiz for somebody who just walked in. Let me see. Are you Brian?
Yes.
Brilliant. Everyone meet Brian. Brian has a quiz. Here you go. We had a longer discussion of these at the start. Okay, so let's plow ahead with our discussion. We're going to start with the electropherogram. Now this is a pretty ancient one. If you have that open on your laptop, maybe you can see this number up in the corner. These date from 1997. Does everyone have fond memories of 1997? I was in grad school in 1997. It was the end of my first year. And the sequencer, the ABI 377, was a pretty big deal back then. I hear that you were 2, that you were 3. I understand. It's all right. Someday, you'll be as old as I am, and you'll be thinking to yourself, well, I remember when high-throughput sequencing was 10 million reads. Those silly people back then. Well, at the time of 1997, I was still thinking to myself, well, there are probably at least 100,000 human genes. Oh, we know that's not true. Imagine what things you think are true right now that science will discover is not by the time you're my age, right? Pretty cool. All right, so here we go. We have an electropherogram. So what we are seeing is time over this axis and fluorescence in 4 different channels shown above that. So an electropherogram is this relationship between time and intensity. Now sometimes these separations are by space rather than time. So in the very old days, instead of using capillary electrophoresis and causing these bases to pass by one at a time, thus being time and intensity, we would separate different length complementary sequences in a gel, and then look throughout the gel to see what color was most intense at each spot. So that would be an example of space and intensity rather than time and intensity. But the concept is quite the same. So as you look across these, you should sort of think to yourself, boom, 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 boom. You've got this sort of heartbeat going on with the electropherogram, a certain frequency with which we see spikes appearing in one channel or another across this electropherogram. And that is one of the clues that we use to make sense of them. So we're going to talk about Fred as a way to estimate base calling errors. So Fred was another one of these algorithms out of the Phil Green group. He also had an assembly program called FRAP. And so you might think to yourself, why does this person keep naming software with PH in his initials? Well, they were his first 2 initials. One of the biggest mistakes I made was naming my first proteomic assembly software DTA Select. Everyone assumed that because the first 2 initials were mine, that I'd named it after myself. I am not that kind of monster of arrogance. I am arrogant, but that was not my type of arrogance. All right, now, we want to estimate base calling errors. So we need to be able to translate this intensity and time into a series of letters. And generally speaking, that's pretty straightforward, right? If at a particular position you have a red peak and nothing beneath it, no black peak there, no green peak, no blue peak. If you just have red there, you say to yourself, that is a T. And you're done, right? Easy. But we need to have a confidence that we can estimate associated with that. What is the probability of error at this one position? This was all published back in 1998 by Brett Ewing. So we want to infer sequence, that's fine and good, but we want to associate a probability of error for this particular letter. Now I am summarizing really, really heavily, and I feel like I'm not doing it proper justice, because this thing operates in multiple phases and it has all kinds of cleverness built into it. But there are various principles at work in how Fred scores are computed. And those are, this peak matches the beat of its neighbors. Remember I was saying that we expect a pretty regular spacing in these electropherograms. This letter, that letter, this letter, that letter, they come at about the same spacing. So we want to be able to infer what the beat of this electropherogram is and say this peak appears right where a beat should be. Next, only one trace is concave down at this call. Ooh. Now, I could throw the term second derivative at you here, but I'm not going to do that. So first derivative represents the change in the signal in response to time. And then a second derivative is a change in the change. Oh, I just went too far with one of the mathematicians in the room. And that's okay. When I say something is concave down, I mean that it forms a shape like this. This is concave down, this is concave up. And you can spot things like that in a second derivative quite easily. So we want to see a place where we have just one trace that forms a downward arc. and that the other traces do not form such an arc at that position. The other thing is that misery loves company. And if we have an oddity at a particular point in an electropherogram, we become immediately suspicious of all of the base calls nearby. So ideally, we want a good sequencing call to be very, very far from any oddities in our electropherogram. Okay, so that is a lot of hand waving, but those are the general principles upon which a Phred probability is inferred. I see some wrinkled brows, so I'm going to pause for a question right here as I drink some lovely green tea.
What is an oddity? Sorry.
No, that's a good question. What's an oddity? So let's look back at this electropherogram I've given as an example. This is a beautiful electropherogram. This is really a lovely one. But there are still some strangenesses to it. So look at, say, position 31. You see how this peak gets cropped at the top? That means that the amount of intensity that we saw for the green channel rose above the maximum we could actually detect. And so we've got some blatting. The top of the peak is not measured because it's lopped off by the limits of the detector. That is bad. Let's look at about 720 out here. You see how this green line is just kind of waving around back and forth through that position? That's a case where the beat is no longer distinguishable. That we're not sure if that's 3 or 4 or 5 different letters all in a row, because we don't have good baseline discrimination between the rise and fall of one peak and the rise and fall of the next. So we could contrast that with, say, the A's that we see around position 305 or so. Here we have green, 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 but you see that it's falling almost all the way down to the baseline after each one. So this is bad behavior. We don't have clear separation of peaks. Here we have very good distinction of peaks. I wanted to look for one other example. It's not really clear here, but I'm going to point to this position around 469. You see these get called CC, but if you look down in the chromatogram, you see that there's actually a fair amount of red signal being shown here as well. That could represent a corruption of the template where you've got some amount of a minority sequence also present in the sample. And that's causing a diminution in the confidence with which we make that call. Yes, the blue is higher than the red there, but there is some red signal being observed, and we don't know quite why. So those are examples of badness. The other thing I would just point out is that at the very beginning of these sequencing runs, we've got a bunch of what I would classify technically as nonsense. And then back here, at the bottom end, we have a real problem distinguishing peaks from each other. Their positions frequently don't separate into peaks, but rather a big glob of red and a big glob of green. So those are the kinds of things I talk about in describing a badly behaved electropherogram. Yeah, if we could get the back door shut, that'd be great. Okay, I hope that helps. Does that explain the concept? Okay, great. The definitions in the manuscript are so much more technical than that, but that's the upshot of what's going on. Other questions? Well, a lot of what I've described sounds good, and it sort of makes intuitive sense, but I haven't told you technically how to go after these things. So I'm going to give you a couple technical details. Now, I am bringing up Fourier Transform because I think Fourier Transform is one of the coolest things ever. It shows up all over the place in all kinds of fields, and therefore I think it's really one of the most central of algorithms that has shown up in bioinformatics. The sort of thing I ask quiz questions about too, so I'll just mention it. So, Fourier transforms decompose a signal to sums of frequencies. Essentially, if you have something and you want to find the beat in it, a Fourier transform is your ideal friend. So imagine that you're at a dance party and you are listening to some house music. I mean, it could be quite that would be cool. But let's just say it's house music. What is the defining characteristic of house music? The beat. Very nice. That was you, Darren. Do you have a big car stereo system or anything? Oh, okay. Well, that's fine. When I was in college and a friend of yours was throwing a noisy party, he was probably playing house music at it. It's terrible music, it's not very insightful, but the one thing you can say is that it has a beat. If your friend was one of those particularly obnoxious people, he probably had one of these graphic equalizers in his system. And so every time there was let's just first get out of our systems, what does house music sound like? Boom, 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 boom. Easy, right? So in the graphic equalizer, you're going to see at the left edge a bar jumping up and down every time there's a big whomp sound coming out of the speakers. And then maybe an electric guitar peels in there with a high screeching solo. And then over at the right of the equalizer, you suddenly see some bars jumping up and down over there. Those reflect that different frequencies are being played in the music that you are hearing. Fourier Transform is one of the ways that we can infer these frequencies out of the sounds that we hear. But I'm not talking about sound now. But the same technology that we use for inferring frequencies to build an MP3 file from a wave, for example, is what we use for inferring the beat in an electropharogram. So instead of having the intensity of sound at a particular moment in time, we have an intensity of fluorescence at a particular moment in time. It doesn't matter to FT. It's going to be able to work with it in either case. So let us imagine then that we have a signal that looks like this. This is the intensity of fluorescence, or whatever. It can be whatever measure we need it to be. It doesn't actually matter. It can be decomposed into frequencies and an intensity associated with those frequencies over here at the right. So I'm going to try to walk you through that. If we just have a sine wave, sine waves just move up, and actually I guess this is a cosine wave, given that it is that has a value of 1 at 0. That's fine. We have this frequency here. If we look at that in Fourier space, if we look at what frequencies are present in it, there's just one frequency, right? Because a single wave can explain this. We have a particular bandwidth associated with it, a certain distance between successive peaks, and it has this intensity associated with it. So Fourier Transform was able to recognize that a sine wave is represented by one frequency at one intensity, and also negative frequencies at positive intensities too. It doesn't matter. So this is a wave that has a lower lower interpeak distance. That's a higher frequency, but it's also not reaching as high or as low as this other frequency did. So this is going to have a lower amplitude. That's the difference between high and low, essentially. And it's going to have a higher frequency than this does. So you see that the frequencies that we see in Fourier space are higher in frequency, reflecting the fact that it's a smaller distance between peaks, and it's a lower magnitude, reflecting that the amplitude here is less than it is here. The magical thing happens is that you can make complex waves by combining these signals. So now we have two things going on. We have the big amplitude, low frequency wave, and the high frequency, low amplitude wave getting combined. And when you sum them together, you get a pattern that looks like this. So you have a little wiggle built into this curve now. Fourier Transform can start from a complex signal like this and decompose it into frequencies that can be added together to produce that complex signal. This is what happens every time you make MP3s from a CD that you shove into your computer. All these different moments in time being separated into frequencies and magnitudes at them. It turns out to be a very compact way to represent sound waves. So, to apply this in the context of base calling, you just need to substitute the volume of a particular frequency for the intensity at a particular time. And this allows us to learn the frequencies at which bases are appearing at this moment in time from the sequencer. Anytime you hear the term Fourier Transform, I want you to think, oh, the frequencies found in X. That's really what I want you to take away from that. Okay, so how is that useful? at the top here, I've drawn in evenly spaced arrows, 4 evenly spaced arrows. Those represent an ideal frequency that's been inferred out of the total signal at this particular moment in time. So we have, we can in effect consider these predicted positions for where each peak should be observed. At each position, we can then ask, what is the shift between the position that we expected to have a big peak and where we actually observed a big peak? If those are very far away from each other, we are less happy with the signal there. If that peak appears right where we expected it, well, we feel a whole lot better about that. So this is an example of the badness that you mentioned a moment ago, that if we have a peak that's very far out of kilter with where it was supposed to appear in time, we are going to attribute it a higher chance of error. And if it's very close to where we expected it to be, we're going to attribute it a lower chance of error. Do people feel good about that? Okay, so when I say Fourier transform, you immediately think, there it is, frequency. Very nice. Great. Okay. The next piece of this is also ridiculously common in bioinformatics. So this too, this is just kind of this is like the part of the talk where I put all my quiz questions. I don't like giving people bad grades. I'm going to help you cheat in favor of getting good grades here. So, negative logarithm scoring. We very, very frequently have the need to represent probabilities that are very, very close to zero in a way that helps us to make good distinguishing, to distinguish well between values that are all close to zero, but some of which are a fair bit closer than than the others. So the probability of base call error is one that is an example of this. If we have a probability of base call of 50% and a probability of base call error of 25%, do we care about how carefully we discriminate one from the other? It's kind of a trick question. You might think that we should, but in effect we don't. The reason is that if you have a base call probability of error of 25%, you're going to throw it out. If you have a probability of base call error of 10%, you're probably going to throw it out. So in general, we don't care that much about having good precision about our error estimation in this higher than 10% error range. On the other hand, we do need to be able to discriminate between saying that something has a 1 in 10 chance of error and a 1 in 100 chance of error. And if something has a 1 in 100 chance of error and a 1 in 1,000 chance of error, we still want to tell those apart. But if you were to plot these on a linear scale, well, they're all just right down here by 0. Relating these numbers by their negative logarithms is one of the ways that we can emphasize the distances between these probabilities when they're all very close to 0. So, if our probability of error is 0.01, that means that we have a 1% chance that this base call is bogus. We would express that as 1 times 10 to the minus second. Everyone knows that when I see a 1e minus 2, I mean 1 times 10 to the minus second power. We've seen this notation. That's great. Okay. So that's 1%. The way that we read that in producing a negative log p-value, in the way that we express a Phred score, which here I'm calling Q, is to take the log base 10 of this probability, which is minus 2, multiply it by negative 10, and that gives us 20. So anytime you see a Phred score of 20, you should think to yourself, there's a 1% chance that this is bogus. If you have a probability of error of 0.001, we express that as 1 times 10 to the negative third. It equates to a 0.1% chance of error, 10 times better than this scenario. We then just grab the exponent, essentially. So negative 10 times negative 3 is 30. So when you see a Phred score of 30, that tells you there's a 1 in 1,000 chance of error. right? So you see how this is flattening out. 1 in 100 becomes a score of 20. 1 in 1,000 becomes a score of 30. A 1 in 10,000 chance becomes a score of 40. So when we were looking at those Phred scores this morning and looking through the FASTQ file, it had already made its assessment of how reliable each one of these base calls was. And it expressed the scores in this negative log space. And there are so many times when we express very, very small probabilities in the space of bioinformatics that negative log notation for reporting those values is commonplace. It shows up all over. So, are there questions about negative log scoring? I'm going to ask another question. If I have a bunch of numbers, and then I take the logarithm of those numbers, do I change the order of those numbers? I see heads shaking. Yeah, you don't. So taking the logarithm of numbers is going to change their relative position, but it's not going to change their ordering at all. It just changes how they're spaced, essentially. Okay, so Q is the Phred score in this case. It uses the negative log notation. It shows up all over the place, so be prepared to see those. All right, now there's a whole bunch of jargon that shows up as soon as we start talking about large-scale sequencing projects, and I thought it was useful to have a little definitions page. So a read, I've already used this term a few times, but I want to reassert that this is a series of letters that has resulted from an individual sequencing reaction on an individual template. So that is a read. So a sequence corresponding to a single electropherogram represents one complementary sequence to this template sequence. The template itself is the piece of DNA that you want sequenced. Now, I play a little loose on the term insert versus template. You may have a long piece of DNA and you're only going to sequence this little patch at this end and this little patch at this other end. So this stuff in the middle is not strictly speaking template because you're not ever sequencing this part. You're sequencing the ends of it. We frequently use the term shotgun sequencing or whole genome shotgun. These reflect the fact that we are going to produce patches of sequence distributed randomly across a larger region. This is something that there was a big debate about in the sequencing community in the early 1990s. People argued that sequencing is expensive and slow, so we should sequence only the fewest number of templates that we possibly can to sequence the whole genome. But the thinking had flipped entirely on its head 1 decade later. Whole genome shotgun had come to the fore, and people said, well, sequencing is cheap, so it really makes more sense for us to just produce a lot of sequence in hopes that we'll get everything covered. So you see this conflict between ideas. Phil Green wrote a really great article about this subject, actually, because the papers that initially publicized the human genome in 2001 suggested that basically everything could be done by whole genome shotgun. But Phil noted that a lot of their success had come from the fact that they had these much longer stretches of sequence that had come from the public sequencing project long before that. So it's an interesting debate. It's very valuable for you to have some sense of how modern sequencers differ from old school sequencers. There may come a time when you order a sequencing project for your own research. Is it the case that a high throughput or next generation sequencer is always superior to an older Sanger style 1990s sequencer? I've already mentioned one way in which the old school sequencers were kind of nice. Long reads. Our ability to produce long reads from old sequencers, the old capillary electrophoresis fluorescence route, was superior in most ways to the sequencers that we use today. There are, as I've mentioned, some sequencers that you can purchase today that also produce long reads like those from Sanger sequencers, but in general, we find that machines like the Illuminas and the Ion-Torns and so on produce relatively short reads, maybe a max of, say, 200 base pairs or something like that, although my numbers are a little out of date. I would talk to an expert on that score. So, first off, the old school 1990s technology generally produced longer reads than do the new sequencers. Sanger sequencing typically gave better base call probabilities than, for individual reads. But high throughput sequencing produces many more replicates of each read. So isn't it valuable then if we can use the duplicate sequencing that we've performed on these highly parallel sequencers of today to boost our confidence in any individual base call? Right? If you have 15,000 copies of a sequence from independent reads, you might think that you can you might think that you can place higher reliability in the sequence you derive from it than if you have just the one. So per read, the Sanger sequencers are in a lot of respects better than the new sequencers. But the new sequencers produce a huge degree of volume. There's a quote I used in a debate tournament, and I caused the judge to roll his eyes at me. I was a terrible debater in high school. But it's a quote, I believe, from Lenin, who was told that the jets the Soviet Union produced were somewhat behind what the West could offer. And he replied, "Quantity has a quality all its own." And that is true of sequencing. We produce so many more sequences from next generation or high throughput sequencers that in a lot of ways we're able to overcome the weaknesses of that technology. Okay, so high throughput sequencing produces sequence from an incomparably larger number of templates in each experiment. Millions, not tens. So if you had 96 capillaries in your old school Sanger sequencer, you are going to sequence 96 sequences at a time. With today's, doing tens of millions is commonplace. All right. This brings us to Burrows-Wheeler. I think that to some extent these algorithms get a little bit beyond what everyone can be expected to understand, but I certainly want you all to understand the purpose of this algorithm. So Burrows and Wheeler are two people who are not known for their bioinformatics skills. David Burrows is a computer scientist of the old school, and he developed a technology that is widely used in practically every programming language today. It's called the subroutine. People like that, people with that kind of stature in the computer science field, have turned their hand at some points to bioinformatics. So his introduction of the subroutine would probably be a much bigger marker on his claim to fame than the Burrows-Wheeler algorithm is. Mr. Wheeler here is he's also an interesting critter. In the days before Google, there was a search engine called Alta Vista, which in some ways prefigured a lot of the advances that Google brought to us. And Mr. Wheeler wrote the Alta Vista search engine. So these are two really great computer scientists who have very long pedigrees who occasionally thought thoughts about biology. So their contribution was the application of a suffix array to the mapping problem. So just as a thought experiment, you have a sequencing read, just one sequencing read. You've got about, maybe it's from this morning's experiment, so you've got 72 base pairs. You want to figure out where in the genome this 72 base pair sequence aligns, right? And let's use the case where we're simply looking for identical matches. I have these 72 bases, I want to find them. So if you as a person were pitched this problem, how would you go about, do you have an answer on this one? How would you go about finding where those 72 base pairs hit the reference genome? I'm picking on you, I know. Why? I don't know. You're just there. You looked like you were following and paying attention and thinking good thoughts. Imagine that I show up to you with a piece of paper saying, here's 72 letters, find them in the genome. What do you do? Okay. Imagine that I have given you a piece of paper with 72 letters on it. This is a sequencing read. And I'm asking you now, where does this match to the human genome? Yeah, so you download a copy of the reference genome. Yeah, it's just this sequence and 23 or 24 different chromosomes.
Yeah, so I'll take what you gave me and compare it to what is the
Yeah.
reference.
Okay, so maybe you open them in a text editor. I don't know, whatever. And then you do, I would probably hit Control F for find, and I'd type in my 72 letters, and I would hope that the 72 letters I input matched somewhere. This is a reasonable algorithm, wouldn't you agree? Okay, that's great. So that works for one search, but how long does it take a text editor to search through, sorry, Marissa, you're gonna have to help me here. How many nucleotides in the human genome?
No idea how many units you know.
Ah, all right, well, we'll make it easier. Mycobacterium. How many nucleotides in the Mycobacterium tuberculosis?
Roughly 4.4 million.
4.4 million. 4.4 million. We'll say it's 4.4 megabytes. You've got a 4.4 megabyte file representing the MTB genome, and you've got a 72 letter sequence. So your text editor takes how long to find that match?
It might take very long. I won't even finish.
Okay, I'm just going to make let's call this a round number. I'm going to call it 10 seconds. It takes 10 seconds for your laptop to find this 72 letter string in the 4 million letters of the Mycobacterium tuberculosis sequence. How many different sequences do we get from a sequencer though? How many different reads? Anyone? Any number will be fine. How many different reads do you get from a contemporary sequencer? Millions, right. Let's say 10 million. Okay, so if you want to search for every read that comes from a modern sequencer, you need to repeat this 10-second search 10 million times, which means 100 million seconds. This is clearly not acceptable. This is a very, very long process, and yet that's what mapping implies. We're trying to map to find the location in the human genome for every read produced by a sequencer. That is a steep challenge. Okay, so Burrows-Wheeler is our way to cheat, to do this really bloody quickly. What it involves is building a suffix array. So a suffix array means that for every string, every substring ending at the same place is also a part of the array. So let's take my name. My name is easy, right? David. Okay, so we have DAVID. That one is going to be in our suffix array. AVID is in our suffix array. VID is in the array. ID and D. Right? That's the suffix array. All right. So our suffix array is going to be set up like this. Burrows-Wheeler adds the preceding letter. So empty set David, D Avid, A VID, V ID, and I D. Those are the different pieces. So you see that the preceding letter shows up here. They use a $sign here to represent that this is the start of the string. And then we have the rest of the string represented here, again with an end of string marker, using a $sign. And we sort all of these. So this, if you want to see the entire string, it's right here. GATG, CGAG, AGATG. That's the whole string. And we can tell that because the preceding letter is the $sign and the final letter is the $sign. That's the full sequence. If we truncate this first letter, we now have this string over here. G is the preceding letter, A, T, G, C, blah, 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 blah. Everyone see that? If you keep knocking off an initial letter, you get this whole array out of one input string. You can then sort this array based on the first letter of the suffix. So $sign represents just the final end of end of string for this. This is the next to last one, the next to last, the anti-penultimate. Sorry, I just love that word, it's a lot of fun. This is a data structure. This data structure is called a suffix array, and it's got this marcation where it shows the preceding letter. Burrows and Wheeler were able to prove that this structure enables an extraordinarily rapid search through a very large string. You could imagine producing a suffix array that includes all of the reads that we have, or all of the sequence of the genome itself, for that matter. So, what they showed was that this structure accelerates the process by which we match to the reference sequence, the genome itself, these reads. And it uses a process of bisection. Bisection is a very important concept. We frequently see it used in binary search trees. So I'm going to try to explain that as I restock my teacup here. Here we go. Ah, lovely tea. Okay. So, how would we do a good example of a binary search? What if I were to seek out a person in this class who is 5'4"? Say I'm looking for someone who's 5'4". Well, first off, we'd have to convert from the metric system because I'm an American. I'm sorry, it's one of those things. But as a computer scientist, if I'm trying to figure out who in this classroom is 5'4", I don't want to just start picking random people. I mean, I picked Gerald. Gerald, are you 5'4"? No, you're taller than 5'4". Are you 5'4"? I mean, doing this one by one has a pretty big miss rate, right? But what if I can get everyone in the room to get an order by height? Okay, so now I've got you all lined up against this wall, shortest person over here, tallest person back here. Now I can use this binary search method to speed things up quite a lot. So I start at the median. I go to right here, the middle person in the room. Who do you think's the middle in the room? Somebody want to volunteer? It's all right, we won't make you stand up. I'm pointing at this person who's standing in the middle of the classroom. This is the person whose height is median for this group. And I say, are you 5'4"? And he or she says, no, I'm 5'6". Well, now I know that I don't have to think about anyone all the way back to that door, because anyone who's on this side of him or her, I forget which I'd said, is taller than 5'6". So I can immediately rule them out. So you see that my search space has just been chopped in half just by using a proper ordering. Next, I'm now going to look at the people who remain in consideration. That person in the middle who is 5'6", that person's out. But everyone below that height is still in our roster. And again, I choose the median of the people who remain in this list. And I say, you in the middle, what's your height? And this person responds, I'm 5'2". Now I know that everyone forward of that person is off of my list. So now I have only 1/4 of the original set that I have to ask. Again, I choose the median, and boom, I finally find somebody who's 5'4". I appreciate that causing students to stand in the order of heights is a substantially easier concept to understand than a suffix array sorted for a Burrows-Wheeler algorithm. But in fact, they both are optimal storage structures for bisection search. So when I have a particular 72-letter read that I want to look for in the genome, well, actually when I have a very large set of 72-letter reads that I want to seek out in the genome, by having created this data structure of them, I am able much more rapidly to find exact matches between these reads and the reference. And that's what Burrows-Wheeler is about. If you look at algorithms like bow tie, which is a very, very popular one that's part of, I forget what we call it, the tuxedo set? I don't even know. Bow tie, cufflinks, top hat, all these different algorithms. The bow tie algorithm is an implementation of Burrows-Wheeler to enable us to very rapidly search for the mappings between a set of sequencing reads and the reference genome. Okay, do people feel good about what the concept of bisection search is? We don't.
I understand your example, but I have difficulty applying it to the sequence.
Yeah. I'm taking you that last mile is probably not the best place for me to operate, I'm sad to say, but I haven't worked with these a whole lot. It's difficult for me to grapple in my mind with why a suffix array is a data structure that enables us to do these searches so quickly, but in fact it does. I would suggest that you read this tutorial on the Burrows-Wheeler indexing method, because it's really, really good on this subject. What was that?
Nothing, sorry.
Oh, okay. Well, the URL I've given here is really, really good. It's one of the best depictions I've seen. I hope that helps. All right. Do people feel good about understanding bisection search? The example I gave with the students. Okay. Good. Well, that is at least a very important concept that shows up all over the place. Okay, on we go. So, now we have done a mapping process. Maybe we even have sort of an error-prone mapping process, where if we couldn't find an exact match for a sequence, we still have an off-by-one matching. Ideally, most of the input data is going to be matched perfectly to the reference. But occasionally, we find sequences in our reads that do not have exact matches in the reference genome. They're mostly matches. They're almost perfect matches, but maybe just a single letter is different than we expected it to be. Maybe where there's a C, we instead had a T. So, how can we figure out whether this happened because of a sequencing error or because this biological sample had a variant at that location? So, we argue several different lines of thought here within the VAR-scan algorithm. This is the paper that announces it. This is where you can download it and look at the source code if you feel like it. So, VAR-scan is intended to separate these random errors that occur by sequencing faults from those that are real sequences. So, we start with the idea that sequence coverage should be high at that position. That may not make a whole lot of sense, but think about it this way. If you have a location in your genome that has a whole pile of reads on it, you know, a depth of 50 or a depth of 100 different reads, your ability to determine whether this variant is consistent across your data is a whole lot better than if you have, say, 2 or 3 or 4 reads at this location mapped to the genome. So we want more data rather than less. If we have fewer reads, the explanation of error becomes more and more likely. Next up, the base calls that declare a change at this position in the genome should have reliable Phred scores. Well, that makes sense. We went to all this trouble back there doing Fourier Transform and all this other nonsense to determine the probability of error at individual base calls. So if we have that information to work with, if you see a Phred score of 20 for a variant call, or a Phred score of 15 for a base call, are you going to feel better or worse than if you have a Phred score of 40 at that base call? Which way is better? This is an important question. In what kind of base would you give more confidence? One of a score of, say, 15 by Phred, or a score of 40 by Phred? 40? I'm hearing 40. What's the probability of error for a base call of 40? You knew I was going to ask that.
1 in 10,000.
Very nice. 1 in 10,000, that's right. All right, so if you have a base call with an error probability of 1 in 10,000, you're going to place it as much more reliable than something that's, say, 1 in 100, which is what a score of 20 accounts to. Very good. Okay, so more data is better. Data of high Phred score is better. a sufficient number of reads must attest to the variant allele. I would note that we generally have 2 copies of each gene. So it may be that if you're sequencing a person and you have a mix of both maternal and paternal chromosomes for a certain location, that you may have a 50-50 mix representing a person's, that you've got 2 copies of each gene. So it may well be that only half of the reads you see for this location show the variant. Or it may be that all of them show the variant. Worth thinking about. So this is the problem of dealing with heterozygosity for a particular variant. Okay, and finally, a statistical test must be used to establish significance. So there are ways that you can use count data as produced in reads to be able to say the probability that this degree of disunity would be seen given the Phred scores and given the number of reads that we have for each of 2 variants. So there are statistical systems built into VARSCAN that enable us to sort of pin down a p-value, saying how likely we would see this variant, a variant this compelling, by random chance alone. Okay. Now I've been talking a lot about sequencers, sequence this, sequence that, and so on. But I feel it is very important to remind us that for a very long time, microarrays were the standard way of determining genetic variability in populations. When the first papers came out suggesting that a $1,000 genome was possible, a lot of people scoffed. They made it sound very much like scoff! Because the idea that you could sequence a person in a day for $1,000 was shocking. We just spent millions of dollars to sequence one person. That was pretty shocking. So, microarrays then became the standard way to measure genetic diversity. They mostly did this by creating SNP chips, and we'll talk about that in more detail later on. But the short version is that we're able to use markers. Well, in the one case, we are able to recognize SNPs in populations by seeing that certain positions in the genetic code have different letters in different groups. So some of this is due to ethnicity, and some of it is due to subsequent mutation and so on that's been promulgated through the rest of the population. So I come from a branch of humanity that wasn't getting quite enough sunlight. And as a result, we weren't getting enough vitamin D, and lo and behold, a mutation in our genomes allowed for us to get more sunlight through our pallid skin so that we would not die of vitamin D deficiency. Right? So this is an example of a genetic marker that I carry reflecting the particular evolutionary heritage of my bloodline. Okay, so these are things that have caused the differentiation of families and of populations on a larger scale. So photolithography has been the primary method used by Affymetrix in producing microarrays. They make genotyping arrays that contain as many as 2.6 million markers. There was a big project called HapMap that was built around the idea of finding as many SNPs as possible across human populations. And so they went through this process of finding individuals from populations all over the world to determine the locations of these SNPs. Now, here's an example where the genetic diversity of Africa has really been kind of passed up. I believe that the Yoruban population in Nigeria was included in the initial work of HapMap. But, I mean, you might ask, what does the Yoruban population have to tell us about the population of Zulus, for example? Maybe not very much. So, we see that there are markers for European populations and so on. You can look up each of these codes within HapMap to see what populations they relate to. But from that set, 2.6 million markers have been used in the current offering that you can purchase from Affymetrix. Now, we talked a little bit about bead arrays this morning from Illumina. And this is a case where Illumina has found a way to measure up to 4.6 million markers in a single array type experiment. Now, they're not strictly speaking microarrays, but they're comparable in the kinds of measurements they produce. In other words, if there's no bead in Illumina that looks for variation at a particular SNP, you're not going to be getting any information back on whether or not that location is present. You won't be getting any genotyping information back on that location. You have to have a bead for the alleles that you want to measure, or you just fail to get any information. And Agilent and Merck also sell microarrays in pretty high volume as well. So even though we think of microarrays as being superseded altogether, there are still plenty of them selling. It's just that a lot of the attention in the research community has shifted to sequencing. One of the things that I learned in talking about this with, in looking through the literature on this subject, the recent literature on this subject, is that microarrays have made a kind of comeback in appearing alongside sequencing data. When you have a genetic variant that's present at very low concentration, sequencing may be able to produce some suggestion that this is a variant there, but there's always going to be this sort of sloppy boundary between things that we know for sure are really genetic variants that are present in the sample, and those that we think are there, but maybe due to low copy number or due to low Phred scores, we don't really know for sure that they're variants. So in a lot of respects, having microarray data gives us the ability to balance this expensive and hard to analyze and hard to store sequencing data against something that's a lot more streamlined. So this is an opportunity for microarrays that if you have sequencing data, frequently having the microarray as well helps give you some ability to counterbalance, to evaluate on another footing whether those findings are real or not. All right, and that brings us to genome-wide association studies. Genome-wide association studies are a little hard to talk about for me. When I was in grad school, people were just kind of getting this notion that you could do things like this and that it would change the world. And now there's kind of a sound when you talk about GWAS with people in our field. So it's a little hard to know how to discuss this. I would start by saying that we are able to measure a far larger number of variants throughout our genomes now than we could in the early days of GWAS, which means that microarrays and sequencing technologies should give us far better insight into which genetic variation is most closely tied to a particular phenotype. In fact, when we look at the findings of prior GWAS studies, we frequently find that the number of genes found to be associated with disease is quite small. Frankly, that the SNPs that we discover are differential between cohorts are generally not that tightly associated with the nearest gene. That's a little like saying, we heard that there was a burglary in Eversdal last night. But does that mean that my house was burglarized? Not necessarily, right? We have a lot of DNA, and genes occupy relatively small spaces within that genome. So if you happen to have a SNP located right here, you may be tempted to say, well, the closest gene is this one, therefore this SNP tells us that this gene is associated with this phenotype. But you'd frequently be wrong to do so. So the interpretation of GWAS has frequently been problematic to say, mechanistically, because this SNP is associated, we now know this gene is that getting this allele for this gene is causative of that disease. The relationship is almost never that clear. The next thing is the odds ratios that come out of these things. You may find that, that somebody who possesses this particular SNP has a 10% greater chance of developing this phenotype. And that's a successful study. But a 10% difference is not that big a deal in some respects. I mean, if I know that cutting cheeseburgers out of my diet on a weekly basis would cause me a lower chance of developing diabetes, well, it's probably a better than 10% chance that I'm going to be able to aid my health in terms of diabetes if I chop out a cheeseburger. But a genetic variant is a smaller amount of information. So you see a lot of people in the States now getting 23andMe assessments of the SNPs that they possess within them as a way of learning more about their biology. And that's great. Frankly, I would like to do it myself. But the kind of information that comes back from GWAS studies like this has been underwhelming in its impact on treating autism or Parkinson's or whatever. So, whenever we talk about GWAS, I want you to sort of keep that duality in mind. So, if we could amass 100 patients with a disease and 100 people who don't have that disease, could we find genetic markers for disease risk? This is ultimately what motivates people to do GWAS studies. So, the very first paper, 2005, made it right into Science, has a whole lot of good stuff in it. And I really want to highlight what they did. For people who are doing the first canonical example of how GWAS works, they did an awful lot right. And that is hard to do, so thumbs up to them. They sought markers of age-related macular degeneration. Okay, so macular degeneration. What is macular degeneration? Where's the macula? Is it my elbow?
Eye.
Eye, very nice, okay. So yes, some people when they get a little bit older develop macular degeneration and their eyes don't work quite as well. So wouldn't it be nice if we could identify people at risk of macular degeneration to teach them very good, healthy habits to protect their eye health? That would be great. So, they had 96 cases and 50 controls, and they used microarrays that measured 116,204 SNPs. Pretty good, right? Well, now we were just talking about microarrays that had millions of markers that they measured. So in the time since 2005, in just one decade, we've gotten 10 times as much information about SNPs from the application of a single microarray. Keep that in mind. These technologies are always on the march. They're always getting better. Using these 116,204 SNPs, they found 2 associations. And lo, the floodgates were opened. The people were off to the races. Now, people in Parkinson's, people in autism, people in tuberculosis, people all over the research communities thought GWAS is the answer. So what did they do right? I want to introduce you to our old friend, the Manhattan plot. Has anyone ever been to Manhattan? No one been to Manhattan? You know, for somebody who travels as much as I did in the States, I really hadn't spent any quality time in New York State. It's kind of odd. Well, I did a couple trips there in my last couple years in the States. And Manhattan really is a shocking kind of place. You might think that the sights and the views throughout Manhattan would be amazing. But in fact, when you're in Manhattan, what you mostly see is storefronts. And you may not even notice that the building you're walking by is the tallest in the state. Right? Because it's all just concrete everywhere you can see. And unless you're doing this all the time, you don't actually notice which buildings are the big ones. You could walk by the Empire State Building at street level and not even know. Weird. So, we call these Manhattan plots because we have all these little skyscrapers that were ranging about us. So, I mentioned a moment ago that they had 119,000 SNPs. What was it? Was it 119? 116,000 SNPs. But I want you to notice that number has dropped to 103,000. They decided that the signal from some fraction of the SNPs was insufficient for them to do differentiation. And they did the appropriate thing. They culled them. They pulled them right out. They said, these SNPs don't have sufficient measurement quality for us to include them. They threw them out. That was a really good first step. Many people in biology have some fraction of their data that's not worth looking at, and they don't throw it out, and as a result, the multiple testing problem hits them even worse. Multiple testing correction is a subject I am very nearly religious about, and we will hammer on this point on Friday because it's just something that you have to know. Okay, so we have, in this case, 103,000 SNPs that pass their QC that are evenly spaced along the x-axis. Here's our x-axis. So I realize the numbers are too small to see back there, but at the far right, we have the number 100,000. Okay? So all of the SNPs have been sorted, in this case, I believe, by the chromosome that they're sitting on, and then the genetic position within the chromosome. So each of these SNPs can be compared to between these two cohorts, 96 versus 50, whatever it was, something on that order. So if you think about the p-values that you get from doing a t-test, if you compare 3 numbers to 3 numbers between 2 cohorts, the best p-value you can get is generally not very low. It's low for the context of what you're doing, but imagine how much more power you get when you have 96 replicates versus, I should know the number, 50? 50, 96 and 50. So this is a pretty well-powered assay, but they're running up against the problem that they're doing a lot of tests. They're doing like 100,000 t-tests, right? That's a big deal. So what we have on this axis is the negative log p-value. Where did we hit negative log probabilities just a little bit earlier in this lecture? Phred scores. Like I said, this is an idea that shows up all over the place in bioinformatics. Negative log of small numbers gives us a negative log score. So each p-value is being shown on the negative log scale along here. And we see that we have a few of these that produce incredibly large spikes, whereas most of them are sitting down here in this lower space. So if this is a log base 10 scale, so I want you to think about what it means for something to be at this level, 6, or this level, 5. What is the difference in probability between a negative log score of 6 and a negative log score of 5 if it's log base 10? People don't think about these things when they're reading these plots, and they really should. All right, the p-value is our estimate that a difference this large could be seen by random chance alone, right? In this case, we're showing them on a negative log scale because we want to be able to discern very sensitively changes between, say, 10 to the negative 5th and 10 to the negative 6th. So when you see that this is a log base 10 scale on which we're plotting these p-values, getting a score of 6 rather than a score of 5 means that the p-value was 10 times as low. The difference between 5 and 4 is 10 times as low. So to get up here into the stratosphere means that we have p-values that are, in this case we see, let's say 10 to the minus 7th versus something that's, say, 10 to the minus 4th. So what is the difference between 10 to the minus 7th and 10 to the minus 4th? 1,000 times. That's right, 1,000 times. So when we look at things on a log scale, we're really separating out very extremely things that are very close to 0. Okay, now, I mentioned multiple testing comparison as a problem. And there's a really important part to this plot that I haven't even touched upon, but I really, really want you to understand it. So you see that there's a dotted line drawn across here, and it has a certain text written on it. It says P equals 4.8 times 10 to the minus 7th. Now, does that apply to the number, or does it apply to the bar? I think it applies to the bar, actually. That's fine. So this represents a threshold. They knew in advance that they were getting themselves into trouble with multiple testing correction. Because if you test 20 numbers with a t-test, and those numbers have no real differences, by random chance, you expect one of them to be a significant p-value below 0.05. Just because you've done 20 tests, you automatically expect one of them to be a positive by random chance alone. They decided to play a rather conservative, to play kind of the most conservative way to protect themselves against making false claims of genes associated with this condition. So they used something called Bonferroni multiple testing correction. Bonferroni multiple testing correction is one of those things that shows up in all kinds of branches. I would really, really, really like it if you understood it. And I'm going to walk you through what they've done. In their decision, they said 0.05 is the p-value threshold that we would accept if we were testing just one gene. We did 103,611 tests. We want to protect ourselves against making even one error. Even one. And so they said the new threshold they're going to use is not 0.05, but 0.05 divided by 103,611. So their critical p-value threshold is this vanishingly small number. And here on this graph, they've shown it. They've shown their p-value threshold. Something must rise above this level before we're going to accept it as positive. And their whole goal here is to protect against making even one false positive call for gene association. Okay, that's the power of Bonferroni. And I have to say, if you don't have enough replicates, Bonferroni is going to kill all of your positives. It's going to take them all away. So it's really very powerful in this case that they had 96 cases versus 50 controls. That's how they were able to get p-values low enough that they could beat a Bonferroni criterion. And that set the standard for how people interpreted GWAS tests ever after. Okay? So Manhattan plots are very important. Introduced right here. You can see how they're interpreted. We have all of our genes and the p-values associated with them on a negative log scale. We have a threshold drawn across here that reflects multiple testing correction. Questions? Okay, great. Now, this morning we took a look at the GWAS catalog. If you are interested in knowing what other researchers in your area have found when they have conducted GWAS studies, there was such a bumper crop of them a few years back that the NIH found that it was necessary to combine all that information together to get people on one page about what studies had been done here. I want to check my time. Okay, we're at 3:30. I haven't run us over anything. So, the GWAS catalog is here for you. I want you to think of it as a resource when you are trying to figure out which genes do I want to follow up and which ones are probably not as significant. The GWAS catalog is a very good resource for you. It's now hosted by the European Bioinformatics Institute. There are a few resources that I keep talking endlessly about. One of them is the NCBI. Another that must make it onto that list is the European Bioinformatics Institute. I talk about Uniprot, etc. All of these sites have a lot of the same tools available. So if you wanted to do multiple sequence alignments, for example, you can do them at many different resources, but EBI tends to host a lot of web tools of this sort. All right, so what is stratification and admixture? If subgroups of a GWAS population differ characteristically in SNP frequencies, these differences bias the SNP p-values. This is called stratification. So that's a little more formal in definition, but let's think back to our age-related macular degeneration population. What if this study had been produced by a consortium? Maybe one site here in South Africa, another site in Beijing, and another site in Utah, for example, in the United States. If they were to combine together data from all three of those populations, they would almost assuredly have fooled themselves of finding SNPs that were significant, but were actually associated with the ethnic groups found in each of those sites. So that would have been a real problem. This reflects the problem of stratification. if people in your subgroup differ in their genes on something other than the trait you're actually attempting to measure, that's probably going to introduce a problem. So stratification is the problem that comes out of that. There was a handy paper from 2011 that gave me this discussion of the problem. So populations that came about by the mixing of two or more distant continental populations in recent history exhibit fine scale correlation among nearby SNPs. So these are SNPs that are very close to each other on the same chromosome in the same band, for example, and segmental scale, syntenic SNPs that have not been separated through recombination. Have people heard the term synteny before? Synteny. Okay. When we try to annotate genes for some new organism, we often look at the organisms that are most closely related to it over evolutionary time, because a mouse and a human, say, have only been separated by, what, 100 million years? What's the current estimate? I don't even know. Let's call it 100 million years for now. So, in 100 million years, the ordering of genes on a particular chromosome hasn't changed that much between the ancestor that we share with mice. So if we see genes in a certain order in mice, we might expect genes to be in that same certain order in the human genome. So those are examples of segmental scale similarity, where the genes have been maintained in the same order. Segmental scale correlation among SNPs is most frequently seen when we have kind of a racially non-mixed population, essentially. So, for example, Finnish folks, people in Finland have been found to have kind of the least interaction with gene pools outside of Finland for a very long time. I'm not sure quite why this is. I mean, Finland is what it is. So when we think about the purity of a genome, when we think about the mixture of a gene pool with external ones, Finland sort of sets the standard as like, this is the gene pool that hasn't mixed as much. On the other hand, think about how many different populations have collided here in South Africa, right? We have the migration of black farmers down the east coast of Africa. We have the Khoi and San populations. We have people brought here from the Malay Peninsula against their will. We have, of course, all the European populations that have come down. South Africa is a collision point for ethnicities over hundreds of years at this point. So when we look at SNPs for the populations of South Africa, we're frequently going to run across SNPs that have not been separated from each other, simply because we find that these SNPs are in correlation with each other simply because they haven't been separated from each other. They haven't had enough time to do so. So doing a GWAS study in South Africa on a general population is quite likely to run into problems involving these problems of stratification. So being able to measure admixture becomes absolutely essential to prevent you from fooling yourself with your own data. That is, after all, one of the secret goals of bioinformatics, to prevent people from hurting themselves with their own data. Okay, so a research group here actually spent a fair amount of time working over data sets from the Cape-colored population. Again, I'm sorry, I'm probably not using the most current language for this group. So here we see that we can sort all of the people in the colored population that were used in this analysis based on how much of the Khoi and San genetic contribution that had been identified. So this is already starting with a bit of hand-waving, because to conflate the San and Khoi populations and say that they're one is already kind of doing an injustice to the way things have worked over the last several hundred years. So we're able to look across this uniform population of the colored population and say what fraction of their genetic variability is explained by this relationship to the ancestral Quaison population. And you can see that there's already quite a variety here. It's not as though there's, it's not as though one set of people have all the Khoikhoi and Quaison mixture. We can also look at what fraction of African genetic variation attribution can be seen, and the amount of European and the amount of Asian. I think a lot of people had actually assumed that the colored population had an extremely strong relationship to the Malays that were brought here forcibly over the centuries. But in fact, there's a relatively small contribution of Asian genetic variants over that time. So that was a pretty cool paper that came right from this very division. So I think it's kind of an interesting read. In total, I think we have to note that sequencing experiments are analyzed through a cascade of bioinformatics steps. Error propagates from each step. This is a closing thought rather than a conclusion, right? But think about all the things that need to go right for us to make sense of high-throughput sequencing data. Base calling needs to have gone well. We need to be able to do our mapping successfully. We need to be able to call variants successfully. These are all very complex steps. So if you simply run the software using the defaults every time, you're not really sure what errors you may be introducing into your data analysis. Very frequently, the results you get from bioinformatics software depend rather heavily on how you had it configured. Even if you're just trying to recapitulate what somebody else did with a data set in the past and publication, they frequently do not include enough information to let you do that. This morning we were looking at some data from Sridhar et al. in 2014. It was a data set from chia. They had done RNA-seq in the plant chia. So when they assembled the software using something called Trinity, they found, I believe, 77,000 different transcripts. When I analyzed the same data with the same software, using the current version, I found 55,000 different transcripts. It's the same software, the same input data, but our numbers are 20,000 apart from each other in the transcript space. So keep in mind that there's an awful lot of processing that has to take place along the way, and mistakes at each step can propagate forward to the next. This point cannot be said often enough, and I want you to really internalize it. Statistical considerations are inextricable from bioinformatics processing. There's a reason why Guillain van der Spey fits inside SATB alongside Gerard and me. He's got very good statistical insights. And frankly, each of us in bioinformatics has had to develop some sort of fluency in the language of biostatistics to be able to make sense of our research. Biostatistics and bioinformatics belong side by side. And I hope that in your mind, you will always keep in mind that relationship. The high-throughput sequencer has completely eclipsed Sanger sequencing and microarray technology. I should really put a big asterisk on that point, because in fact we still feel that microarrays can allow us to do a lot of things a lot more straightforwardly than we can do with the current sequencer. Similarly, to say that the Sanger sequencer, the use of capillary electrophoresis and fluorescence to get our electropherograms, to say that that's completely gone is not really sensible. There really are cases where it's a whole lot more convenient to Sanger sequence a small batch of samples rather than going the high throughput sequencing route. If what you're interested in is a relatively small genetic region for a relatively small number of samples, doing it by Sanger is actually much more sensible in a lot of ways. All right, so that's our discussion of genetic diversity. Tomorrow we will come to kind of the interesting world of genome annotation. When you're sequencing something that no one has sequenced before, how do you make sense of it? I find that to be a really fascinating problem. So tomorrow we'll have a discussion of that part of the genome sequencing and annotation pipeline, and then we'll move into the fine realm of gene expression on Friday. So I will see you tomorrow morning at, what time? 9:30, when we will have our next quiz. It's actually written this time, so we're in a much better shape. So I'll see you all tomorrow morning at 9:30, and I look forward to it very much. Thanks.